C-banding of chromosomes. C-banding of chromosomes is a procedure, or it is a method which is used to stain the constitutive heterochromatin. Constitutive heterochromatin is the type of the heterochromatin that is usually lies near to the centromeres. In this diagram, we can see the chromosomes, they have been stained. The pattern of banding is called as C-banding. In this diagram, we can see that as now we know that, let's say this is the P arm of chromosome, this is Q arm of chromosome, and in the middle there is centromere. So C is for the centromere. So when we stain the centromere of the chromosome, the banding pattern is called as C-banding. C is actually for the centromere. Here we can see that all the chromosomes which are present in this slide, they have stains or dark stains which actually represents to the centromere. So here is the centromere, this is centromere of chromosome, this is also centromere. So centromere of the chromosomes that have been stained, and in the centromere of the chromosome there is heterochromatin. What is the procedure of C-banding? How we do the C-banding in case of while we have to do the C-banding of the chromosomes? So this is the complete procedure of the C-banding, in which treat the slides that have been prepared. Those slides, they are treated with 0.2 normal HCl solution. That is usually for 1 hour. Once we treat the slides with HCl, then we rinse the slides with the deionized water. That is the second step. Once we rinse them or wash them, we immerse them in 1% barium hydroxide for 50 degrees at 50 degrees centigrade for 5 to 15 minutes. Once we complete this one, we immerse them in barium hydroxide. We rinse the slides with deionized water. Then we incubate them at 60 degrees centigrade in salt sodium citrate solution. That is salt sodium citrate solution buffer for 1 hour. So this is the procedure how when we have to perform the C-banding of the chromosomes. In the next step, rinse the deionized water and stain in 4% Giemsa stain for the 90 minutes. Rinse the deionized water, dry, and examine the slides under oil immersion. So this is procedure of C-banding, how we do the C-banding. As a result of the completion of this procedure, when we will see the chromosomes under the microscope, we will see that the centromere of the chromosome has been stained. Because the centromere of the chromosome has been stained, that is the reason this type of the staining is called as C-banding of the chromosomes. So C-banding is the banding process of the chromosomes in which we stain the constitutive heterochromatin. Constitutive heterochromatin usually lies in near or in the centromere of the chromosomes.